Hey, this is Ethan, and this is the third video in my series, and today I'm going to be talking about the Infinium assay for large-scale SNP genotyping and the applications and why we use this. The first step is to set up the samples. This is first done by denaturing the DNA, and then incubating and amplifying it, and then fragmenting it. After this step, the DNA is precipitated with an isopropyl solution, and then put into the centrifuge in order to precipitate DNA pellets. As seen in the next slide, you can clearly see little blue pellets at the bottom of the plate. It is important at this stage to let the plates dry, ensuring that there's no liquid left in the plate. After this takes place, there are more reagents added. The samples are vortexed, and the samples are also added into the centrifuge again, as well as the incubator. Finally, hybridization can begin. This is first done by putting the samples on the heat block and denaturing the samples. Next, the bead chips are unwrapped from their packaging, and there's a series of processes of putting each one of the bead chips into a hybe chamber, which includes 4 of the chips. After this is done, there is a solution placed onto the hybe chamber, and the packaging of the bead chips is set off to the side. After this, the DNA is carefully put into the inlet on the arrays. After this, the chips are placed into the T-can where the staining and extension occurs. This process takes a while. Now for the messy stuff. The chips are now washed and sealed, and then put into a vacuum in order to dry. Finally, the chips can be placed into the iScan. Now for the most important part. Here you can see the nucleotides fluorescing that were attached during the extension and staining steps. Later, the intensity and color of these can be used to determine the alleles present at the SNP site. So why is this important? Well, first off, we can plot the intensity data on a graph to see the allele frequency, and this helps us to better determine genetic markers that are associated with certain diseases such as autism. Thanks for watching. I hope you enjoyed.